I mean, this goes back to the work of Otto Warburg in the 1920s and 30s and 40s, where he defined that cancer originated from damage to mitochondria. And that then elicited a whole series of changes, forcing the cell into a fermentation mechanism to survive. And we have validated and confirmed Warburg's original finding. So in order to do that, what I did was to go through the scientific literature, looking at electron micrographs, the very high magnification of mitochondria in tissues, because you can't see them under light microscopy. In order to look at the structure of the mitochondria in the cytoplasm of the cell, you really need electron microscopy, scanning electron microscopy. And then when you look at, so what I did is I went and I looked at all the major cancers, probably representing 95% of all cancer deaths caused by these kinds of cancers. And I went back through the 50s and 60s, 1950s, 60s, 70s, because in a lot of medical schools, people would be looking at cancer tissue with electron microscopy. And then you go to those papers and you look and see what they found with respect to the number and structure of the mitochondria in those tissues. And invariably, they were abnormal structurally. And even if one were to isolate them and look at the biochemistry, it was abnormal. So we have never found normal mitochondria in any kind of a major cancer. Okay, so if you don't have normal mitochondria, that means the cells are not going to be able to generate energy through normal respiratory systems like oxidative phosphorylation.
Cellular respiration. There are two kinds of cellular respiration, aerobic and anaerobic. Aerobic respiration is the process by which oxygen-breathing creatures turn fuel, such as fats or sugars, into energy by the help of oxygen. The key organelle inside cells of aerobic respiration is the mitochondria, where the glucose molecule from the food goes through a multi-step process, such as glycolysis, Krebs cycle, and electron transport chain. At the end of this multi-step process, one molecule of glucose, with the help of 6 molecules of oxygen, can produce 36 molecules of adenosine triphosphate, or ATP, along with 6 carbon dioxide molecules and 6 water molecules. This makes aerobic respiration a very efficient process.
And this is exactly what Warburg said. He said mitochondria oxidative phosphorylation becomes irreversibly damaged in all cancers, regardless of where they come from, thereby forcing them into a fermentation mechanism. And that's what the characteristics of all cancer cells are. They ferment. And then what we did is Warburg knew glucose was the lactic acid fermentation derived from glucose was the major fuel at that time. We have now defined glutamine as a second fermentable fuel. The field had thought for many years that glutamine was being respired. No, it's not respired, it's fermented. So the two fermentation fuels that drive the majority, if not all cancers, are a sugar fermentation and an amino acid fermentation. And without the glucose and glutamine, no cancer cell can survive. So our goal is to scientifically validate fermentation mechanism for a glutamine and show how Warburg was right on his original description, but he also did not have new information which would clarify and resolve this entire cancer issue.
So, Thomas, can you please tell me that role that the mitochondria play in the development of cancer?
They are a second living organism inside a cell. They actually have their own genetic material, and they are the organelle that breathes the air to make the energy. So, what Warburg found was that all normal cells have these organelles that take in the oxygen, and the cancer cells have a defect in that organelle. So they take in less oxygen and they produce lactic acid, which is a waste product of glucose fermentation. They call it lactic acid fermentation. And that proves clearly that these tumor cells are not getting energy through any kind of an oxidative mechanism. So as long as they have the fuel to continue their fermentation, they will survive. So all cancer cells, and every one that we've looked at, they have damage in their structure of that organelle. That means that all cancer cells, regardless of what you want to call them, are all fermenters. They're using an ancient fermentation pathway to survive. So that means that there's a very simple way to kill them. You just have to take away their fermentation energy and feed them things that they can't use. Fatty acids and ketone bodies are not fermentable. You need oxygen to get energy from ketone bodies, and the cancer cells don't use oxygen. So it's a very clear way to kill the majority of cancer cells. And then the amino acid is also, glutamine, is also the second fermentable fuel. Glutamine is the most abundant amino acid in our body. It's everywhere. So in order to, and besides, our immune system needs glutamine, our urea cycle needs glutamine, our gut cells all depend on glutamine. So if you go too aggressively on attacking glutamine, you also can cause metabolic disturbances in the body. So that's why we developed the press pulse theory of strategy. We can control glucose constantly because glucose is a non-essential metabolite. Our body does not need glucose. We can make glucose from within at very low concentrations. But the glutamine issue, you have to get the body into a ketotic state, and then we hit the glutamine just briefly, boom, wait, bing, so it's pulsed. It's like a pulsing. And that just, I have a few cancer cells here, they go down. Okay, then we let the glutamine come back, but not too many come back, just a little bit. Then they hit down, come back a little bit, hit down. And the next thing you know, gone.
As somebody who got one of these diagnoses and found myself searching the tomes of YouTube for information and insight and hope and knowledge, it's very difficult to parse truth from fiction. And there's reason for that, beyond the fact that, science is not always easy to understand and we're not always trained for it. We're at an inflection point, a sort of an overlapping point in history right now, where an enormous group of people, doctors and oncologists, still live in a world where they believe that cancer is a genetic disease, where another group of people have realized and proven and shown with exhaustive studies and research that it is in fact a disease of mitochondrial dysfunction. And though that is the good news, that we now know that we live in this overlapping inflection point, is also the bad news, because you're going to go out there, you're going to watch a podcast like this, and you're going to learn the information we're telling you, and you're going to go and read Seyfried's book or paper or watch his videos. But you're also going to come across an enormous amount of information that literally says the opposite, or that deters you from doing that. And so I hope that a video like this and a story like mine and the stories that you've heard from the doctors Howard and from Daniel can help to point you in a direction that is going to deliver you to a place where you can access truth and not be duped. This type of media is incredibly important, specifically for that end. Understand, I live in a world where we understand what cancer is, and we've been able to apply that science to therapies that can resolve it. And all you need to do is to read that science to join us. And in the future, everyone will be using metabolic therapies. Cancer will not be the horrible death sentence and the terrible tragedy that it has been for the last however many hundreds of years. We're just happy to be a part of making that happen. But you need to understand that it's going to be confusing out there as you read and look and hear what people are telling you to do. But please, if my story can be anything, let it be proof that Thomas Seyfried is right, that metabolic therapies work, and that, the outcomes that you're staring down the barrel of don't have to be the way it is.